Well, hey biology, hope you're doing well when you watch this video. We're going to start chapter 9, the cell cycle. Thank you again for your time investing, your hard work. When you think about science, there's a ton of cycles in science. You have the water cycle, which of course this is an image from probably elementary school. You have the nitrogen cycle. You think about cellular respiration, photosynthesis. You have the Krebs cycle and Calvin cycle. You even have cycles within your own body. The circadian rhythm cycle. Sometimes we can just wake up without an alarm clock. Sometimes we fall asleep roughly the same time each night. Your body definitely goes through a cycle each day. Here's the cell cycle. Okay, this is what we're going to be talking about today. And so the first part of the cell cycle is interphase, which is where a cell spends most of its time, G1, S, and G2. And then you have mitosis, which is PMAT, which we'll get to in a second. And then cytokinesis. So mitosis is the division of the nucleus. Cytokinesis is when one cell becomes two in the division of the cytoplasm. And so if you think about that, why should you care, right? Well, healing, repair, replacement happens with the cell cycle. Growth happens in the cell cycle. When the cell cycle is dysregulated or has possibly mutated, you can develop cancers, tumors, growths. Some organisms actually reproduce through the cell cycle, asexual reproduction. And so you all start like this, one single fertilized egg cell called a zygote. And through the cell cycle, your tissues differentiated, specialized, and you became you. Of course, with a biblical mindset, God, 1 Psalm 139, you were fearfully and wonderfully made. God does not make mistakes. This is also important because tissue needs to be repaired. And so, when there's disease of tissues and they can't get nutrients because of blockage, that tissue dies. And so that is also a reason why to care about this. Hey, another reason, cancer, right? So here's the cancer rates from a few years ago according to the cancer.org, and you can see 1 in 2 males will get cancer from birth to death. 1 in 3 females, roughly. Breast cancer is very high in females. Prostate cancer, very high in males. Those are sex-specific cancers. Lung cancer is, of course, crazy high. So as we go through, make sure to fill out the notes. So why don't cells just grow larger and larger? Well, the larger a cell becomes, the more demands the cell places on its DNA. So cells don't grow as large as they want to. They actually divide when they become a certain size usually. And so this is what it terms a surface area to volume ratio. Okay? And so you want a ton of surface area because you want to get materials in and you want to get materials out. And so volume, so the cell's really big like this, right? It's going to take a long time to get materials to that deepest part of the cell. Also, the cytoskeleton has more demands, DNA has more demands, and it just doesn't work out. And so cells want to have a lot of surface area for volume. Okay, they want to have a lot of places for diffusion and transport to happen. And so here's a couple of terms that when cells are large, that it's just inefficient. Diffusion, the cytoskeleton, which are moving things. Asexual reproduction is the production of genetically identical offspring, so they're clones, right? Sexual reproduction, of course, which we'll talk about a little bit later, genetics, it's a beautiful topic, is when you inherit some of the genetic information from your mom and dad. So here's the Venn diagram about asexual and sexual reproduction. Underline what I underline, but one parent to two, of course. Cells divide by fission, budding, or fragmentation. This is meiosis, which creates sex cells, sperm and egg. Very little. I mean, we're creating clones here. And of course, genetic variation is very important. Here's what's in common with them right there, okay? Usually these are found in more simple organisms, and usually these are in more complex organisms. But life is complex in itself. So clones, yeah. Clones are exact copies, genetic copies. And so identical twins, yes, they're technically clones, but they look a lot different when they get older because of how they live their life. Guaranteed, if you've eaten fast food, you've probably eaten cloned meat. Companies clone whole herds of cattle, cheaper, easier, less stress, more financial revenue. It's just kind of crazy to think about. Chromosomes. Humans have 46 chromosomes, okay? 46. You get 23 from mom and 23 from dad. And so every cell must copy its genetic information before. So let's say you scrape your knee. You want to make more skin cells. Well, those skin cells have to separate and replicate and divide. And so you have to replicate your DNA because you want an entire book of DNA for every cell, okay? So each daughter cell gets its own chromosome number. And so if you want to pause here and see if you can think which one has the most or the least chromosomes, try to order these from, okay, this organism has the most chromosomes to the least. Go ahead and pause. All right, so here's the answer, right? Shrimp have the most, followed by sweet potato, followed by the turkey, dog, human, the 46, and oak tree. And so what I want you to write on your paper somewhere is the phrase chromosome number does not equal complexity. Of course, we are the most complex organisms on the planet, not even close. And just a testament to how we were made. Prokaryotic chromosomes are usually a single circular DNA molecule. So it's just usually one chromosome, not linear ones like we have eukaryotes. And so another reason to care, here's just another reason to care when the cell cycle doesn't happen correctly, and this is of course talking specifically about meiosis, you can sometimes have something called non-disjunction when chromosomes don't separate correctly. And this is actually Down syndrome when you have 321s. Chromosomes were numbered based upon their size, so you have 1 through 22 are the autosomes, and then X and Y are the sex chromosomes. So this is a guy's karyotype, this is called a karyotype, and this is a Y, Y makes guy. And so 21, no, 321s, it leads to Down syndrome. Fully human, we're more alike than different, created in the image of God, but nonetheless, it is another reason to care about this stuff. So you do need to know how chromosomes are organized. It starts with DNA, then they fold around histone proteins, then those kind of join together to form nucleosomes, and you kind of have like this old telephone cord, which leads to a duplicated chromosome. Notice something though right here. Let's say that you see how this term sister chromatids, this is a common misconception in biology. Let's say an organism has 1, 2, 3, 4 chromosomes, okay? When those chromosomes duplicate, oh, it just erased. Nice. 4 chromosomes. When those chromosomes duplicate, they should look exactly roughly the same, right? And then during prophase, which we'll get to in a second, they're joined at the place called the centromere. That's a vocabulary word. But here's the thing. When they're joined, you still only have 4 chromosomes. 1, 2, 3, 4. But you have 8 sister chromatids, okay? So you still have 4 chromosomes because they're joined. But when they separate, when the sisters separate, they are now called chromosomes and not chromatids. I'll show you more here in a second, okay? And so here's just another picture of DNA. And so during the cell cycle, a cell grows, prepares for division, and divides. Bacteria do something like this. They don't do mitosis because they don't have a nucleus, but bacteria reproduce something called binary fission, okay? And so here's just another diagram of the cell cycle. You can see you have G1, which is where a cell grows. S, when the DNA is duplicated, okay, chromosomes, DNA. 46 is humans. And then G2, there's checkpoints. The cell's also growing and making sure it's ready to go into mitosis. Mitosis, if you want to write it down, PMAT, prophase, metaphase, anaphase, and telophase. And then cytokinesis is when one cell becomes, well, that's a horrible arrow, 2. Okay, one cell becomes 2. And so that leads us to the cell cycle, the stages of interphase. Interphase has 3 substages, G1, S, and G2. During G1, the cell is growing and carrying out normal cellular functions, and we'll talk more about that in a second. S is when it copies. G2 is when follows the S phase, when the cell prepares for the division of the nucleus, and then mitosis is the division of the nucleus, and cytokinesis is when the cell divides into 2 daughter cells, okay? So this leads us to right here, G1, S, and G2. So I want to give you a little bit more specific stuff. And so, let me see if I can do like a little highlighter thing. I'm not exactly sure if I can. Yeah, there we go. Nice. Maybe not. Okay, here we go. Growth. Whoa. Growth. Okay, cells grow in G1. They're getting bigger. Okay, they don't get as big as they want to. The S phase, of course, stands for DNA synthesis, as the name suggests. G2, we're making proteins and RNA to get ready for division. But look right here, proofreading. Subsequent repair. We are double checking to see if everything is lined up correctly before we divide. Of course, errors in division can lead to cancer, can lead to problems, okay? So here's the two pictures of mitosis, which I'm going to go into a lot. You can see, for this class, you don't have to worry about pro-metaphase. That's more of like an AP biology thing, so you don't have to worry about that. But you do need to know that interphase, G1S, G2, okay? Centrosomes are these organelles that spindles grow from, centrioles. You see the chromosomes duplicate so that you have your sister chromatids, and then the spindle begins to form, and the spindle attaches here. So as the spindle attaches to the centromeres, technically kinetochores, you don't have to know that term, but it attaches to the chromosomes in prophase, okay? Metaphase stands for middle, so you can see the chromosomes line up in the middle of the cell, and then A for away, anaphase, A for away, and so sisters are pulled to opposite poles. So this is when the sisters now are called chromosomes. And telophase, the nuclear envelopes are beginning to reform. You can see the cell is pinching together in preparation for cytokinesis, okay? So prophase is the first phase. Sister chromatids are structures that contain identical, right, same copies of DNA, and they're joined at something called the centromere, okay? Prophase spindles keep going, connects them, okay? This is what it looks like. Second phase is metaphase. It's a short phase because they line up, again, M for middle. They line up in the middle of the cell, okay? There's metaphase, what it looks like underneath the scope. A for away, so anaphase. Notice how the term changes. You go from sister chromatids when they separate, and after they separate, they're now called chromosomes. They're called daughter chromosomes, okay? This is anaphase. Telophase is when two nuclear membranes begin to form. There you go. And that's what it looks like. But notice something right here. You can see something right here developing in a plant cell. See, plant cells don't pinch together. They have to kind of, they form something called a cell plate. And that cell plate becomes a cell wall. And so that is why it's important, because the cell plate forms right there. Cell plate forms. And that allows the two plants to begin. Again, why is it important? Here's your 4 blanks. Reproduction, asexual reproduction. Not technically mitosis, though, for at least prokaryotes. That's just a cell cycle. Growth, replacement, your intestines, blood cells. I mean, everything needs to be replaced, your skin. And then repair. Yes, of course, we do live life. We get scrapes and bruises. There's organization to this. We looked at the cow eye, right? Well, how many tissues did we see in the cow? We saw 4, right? Well, cells, when they're doing the same function, make up tissues, same general structure and function. Tissues make up organs, and organs make up organ systems. If you want to, you can pause right now and try to identify how many organ systems you could possibly name. Organ systems such as, you can see such as respiratory system, circulatory system, cardiovascular system, lymphatic system, all that sort of stuff. I'm going to talk about cell differentiation here in a second and stem cells and stuff. We're going to talk about that in a little bit. Okay, so this is where honors separates from on level. Regulation. Why doesn't cells just divide all the time? Okay, well, if you want to look up some videos here, human growth hormone is an interesting video. I'll try to show some of these videos in class to you when we're discussing questions and stuff. But human growth hormone causes cells to divide. Robert Pershing Wadlow is the tallest man to ever have lived, known as a gentle giant. He had actually a tumor of the pituitary gland, I believe, which caused an abnormal number of amount of HGH to be formed. Thereby, he was still growing when he died in his 20s, which is crazy. But the cell cycle is controlled by regulatory proteins. So I want you to think of a light switch. Okay? And I'm going to try to draw this. But you have the on side, you have the off side. Okay? You have like your little switch thing, right? And so regulatory proteins determine if a cell is going to divide or not going to divide, okay? So there's things called cyclins, okay? So cyclins are proteins that bind, which means join, to enzymes. Enzymes are just proteins again. Cyclin-dependent kinases, okay? These are known as CDKs. They are dependent upon cyclins. Well, so here's the thing. These cyclin and CDKs, they have to be around for a cell to continue progressing. Now, you see all these stoplights? Oh, all these stoplights are different checkpoints. And these checkpoints, without these cyclins and CDKs around, the cell will pause. It will stop. It will go into like G0, which means like a pause. Like, wait, something's not right. Something's going on. And so the concentration of these guys control different activities. Okay, so the cell cycle has a built-in checkpoint system that monitors the cell cycle and can stop if something goes wrong. Okay, and so I'll go over this example with you. This is actually psoriasis. But for example, yeah, I mean, bone cells aren't growing all the time. But bones do grow when the correct signals are around. I'll go over that with you later on. And so sometimes normal cell growth, they stop dividing once they touch each other. Contact inhibition. Cancerous cells don't care. They grow on top of one another, forming tumors, possibly. And so there's different checkpoints, okay? And so here's just the different types of checkpoints. You have, we ready to go into mitosis? Yes. Have the chromosomes separated correctly? Yes. Are we big enough, strong enough, everything ready to replicate DNA? Okay. Is the environment favorable? Okay. Here's just an example of one. This will be extra credit on the test. But P53 is known as the guardian of the genome. I'll also show you this video in class. And notice something here. If P53 is around, it's called a tumor suppressing protein, which means it stops cells from dividing correctly when something's wrong. Okay? So it's located in the nucleus of cells throughout the body, and it attaches to DNA. So literally, if DNA in the cells become damaged, the protein plays a critical role in stopping something. So watch. This means block. It blocks this, it blocks that, and the cell goes into G0, which means arresting. Okay? It can't progress because these cyclins aren't around. P53's blocked them. And so because of that, the cell no longer progresses into the cell cycle, which means it enables the cell to repair the broken DNA or anything like that. It's essential for preventing tumor formation and literally has been nicknamed the guardian of the genome. So here's an example of cyclins, right? So these are two proteins, protein 1, protein 2, cyclins right here. Notice that this protein is inactive, but since these guys are around, they activate it, which means turn it on, right? Flip that light switch on, and now that protein does its job and the cell cycle continues. It does its job, okay? And that job could be multiple different things. And then it disintegrates, right? Cyclins are destroyed. So that's an example of how the cyclins and CDKs affect each other. And so you can see cyclin concentration increases, mitosis, and decreases. And it builds. And so this is all about signaling, okay? Of course, you can see right here, tons and tons of steps. You don't have to know anything right here. But just see, growth factors, important. This is also a growth factor. Important, right? Turns these cells on to grow more and more and more. So cyclins, just a definition here, just basic understanding right now. Cyclins are a family of proteins that regulate the timing of the cell cycle. They have to be around to regulate and keep the cell cycle progressing onward. Lots of different kinds of cyclins. You can see G1 kind of progresses all the way through it. Cyclin E is important to making sure that S phase happens. Cyclin A is important when it comes to making sure mitosis, as well as cyclin B. And so there's regulation inside, outside, and growth factors. Okay, I would know these definitions. Internal are proteins inside the cell. External ones are, of course, come from the outside world to see if the cell's okay. And growth factors are external regulators that stimulate growth. Example, HGH. So when a cell's not doing well, it goes into apoptosis. Apoptosis is an example of programmed cell death. So when cells aren't doing well, they go into apoptosis. Apoptosis is natural, normal. It's normal during development. You have 5 fingers because of apoptosis. But this is crazy important. Cancer cells avoid this, and we'll talk about that here in a second, okay? So apoptosis plays a role in development of structuring tissues. Here you go. There's your fingers. These cells are actively dying in development of these digits, okay? And then finally, the last part, cancer. How is cancer different? Well, cancer is a disorder when a body loses its ability to control the cell cycle, okay? So this is how cancer cells are different. Number 1, they're not specialized. Sometimes they're even immortal. They have abnormal nuclei, maybe enlarged, maybe abnormal number of chromosomes. They escape undergoing apoptosis. They should die. They don't. Form tumors, of course, most cells stop growing when they touch each other. They undergo metastasis, which is spreading to other tissues, and angiogenesis, which is kind of a really sinister way of basically hijacking your blood vessels, making more of them so they get nutrients and oxygen. Pretty crazy, right? And so cancer is a really hard disease. Of course, we'll all be affected by it one way or another. And so cancer cells versus normal cells. And then benign is non-cancerous. And malignant, don't ever want to hear that term, is cancerous. Okay. And so metastatic cancer is when the cancer is spread. It can travel to other parts of the system, lymph vessels, like blood vessels, go from the primary site to other parts of the body. For example, melanoma could go from the skin to the bone, to even to the lungs, various places. And so that is a problem. Okay, and this is just how it spreads through the lymph system. Cancer cells can go through several steps and does that. So cancers are caused by defects in genes. So how is it caused? Well, cancers are caused when genes, DNA segments that regulate cell growth and division, are messed up. Okay? And so some sources of gene defects are smoking tobacco, radiation, like UV light. Hello, Floridians. Defective genes, viral infection even. Yeah, HPV, crazy high. Even a P53 gene. Again, we can look at that later on. BRCA1 and BRCA2 are human genes that produce tumor-suppressing proteins. When these genes are altered, higher risk of breast cancer. Okay? What are some treatments for cancer? Surgery, radiation, chemotherapy. A beautiful promise from God's word. He wiped away every tear from their eyes, and there would be no more death or sorrow or crying or pain. All these things are gone forever. Cancer never has the last word for a believer. God is God, and God will always be enough. Every tear will be gone for the believer. It's a happy thing to know. All right, and so this is the human body contains hundreds of different cell types. So how do they go? How do they become specialized? Well, during development, cells differentiate, which means specialize, right? Right now, you probably play like 10, some of you might play 5 different sports. But let's just take baseball. Baseball is my sport. Everybody pitches, everybody can play all the positions. But when you get up into that upper, college, even pros, you start to specialize in one position unless you're Shohei Ohtani, I guess. But you specialize in one position, right? And so that leads us to, you just start as one cell. Plant cells have specialized tissues in their roots, stems, and leaves. Cells that store, cells that transport, cells that carry out photosynthesis. All those cells started with the same DNA. How are they different now? Well, that's the process called differentiation. They start expressing different proteins. They become specialized. They can carry out the jobs necessary for only their specific part. And so this starts with stem cells. Stem cells are kind of like blank slates. They're unspecialized, which means they differentiate, and they're known as stem cells. Okay? And so stem cells have the remarkable ability to develop into many different types of cells in the body. When a stem cell divides, each new cell has the potential to remain a stem cell or become another type of cell. Why are they important? Because they're capable of renewing themselves through cell division. And number 2, they can become induced to produce tissue or organ-specific cells, which is crazy to think about it. Biologists say that such a cell is totipotent, literally able to form anything. Why is this important? Well, let's say you need a new heart. You could take stem cells and possibly grow a new heart in a chamber. Specialized chamber, of course, with external growth factors and all sorts of the right recipe. And that's pretty impressive, right? So stem cells, unspecialized cells that can develop into many different things. Adult stem cells, say totipotent, happen really early on in life. But adult ones, the ones that we have, multipotent, which means they can produce many different types of cells. And so here's just a little picture right here about that, where we'll end today. Totipotent, capable of forming 200 different cell types in the human body. That's where the blastocyst comes from. Pluripotent, capable of generating all tissue type, and multipotent, only specific lineage. But there is some really great research, and one got a Nobel Peace Prize, that says basically what he found is that iPS cells induce pluripotency. He can turn any body stem cell into one that can produce any different types of cells. But I won't get into that right now for time, okay? So I hope this was helpful. Again, we're going to be going through questions. I'm going to be tagging like higher level thinking questions on for you. And I'm just going to leave you to this and be like, hey, good luck. We're going to watch some videos, go over some stuff. But just in order to save some time, I wanted to do this. Try to catch up a little bit to where we need to be for the year. Proud of your hard work. Let me know if you have questions. God bless.